Welcome to my channel Biochem by World. In this video, we'll be discussing protein digestion. So first question comes, where the protein digestion occurs. The digestion of the protein occurs in the gastrointestinal tract. So what are the organs which are in the digestive system? The digestive system of humans include the stomach, the small intestine, the large intestine. So let us begin with the topic, that is the protein digestion. The entry of the dietary proteins in the stomach stimulates the gastric mucosa to release hormone gastrin. The released hormone gastrin, in turn, it stimulates two types of cells of the gastric glands, that is parietal cells and chief cells. The stimulated parietal cells, they are releasing the hydrochloric acid into the stomach, whereas the chief cells are stimulated to release a zymogenic form of enzyme, that is pepsinogen. As I already told you in the last slide that the entry of the dietary proteins in the stomach, it stimulates the parietal cells of the gastric glands to release hydrochloric acid, which results in the decrease in the pH in the stomach, that is the pH of the stomach is 1 to 2.5. What is the function of this acidity of this hydrochloric acid in our stomach? First, it acts as an antiseptic, that is, it kills most of the bacteria and other foreign cells which accompany with the food we eat. The second important function of the hydrochloric acid is it acts as a denaturing agent. It means that it unfolds the globular proteins which leave the internal bonds, the internal peptide bonds, more accessible to the enzymes which are there for their hydrolysis. The third function of the HCL is that this low pH, it helps to activate the zymogenic form of pepsin, that is pepsinogen, which is released by the chief cells of the gastric glands. The low pH activates the pepsinogen to pepsin. On the other hand, pepsin, that is activated by another phenomena, that is autocatalytic cleavage. What do you mean by autocatalytic cleavage? Auto means on its own. That means that activated pepsin, it goes and cleaves its own zymogenic form, that is pepsinogen, activating it. Alright, that is. So there are two ways that is pepsin is activated. First is the low pH, which is given by the HCL in the stomach, and the other one is autocatalytic cleavage, that is on its own. It cleaves its own zymogenic form when activated. What does pepsin does? The activated pepsin, it hydrolyzes the amino terminal site of aromatic amino acids. As you all know that proteins have two, we can say, ends, that is first end is the amino terminal, that is NH2 amino terminal, and the other is carboxyl terminal. Alright, it has two ends. So suppose here there is aromatic, and there are three types of aromatic acids, that is phenylalanine, tryptophan and tyrosine. Suppose phenylalanine is there. So what pepsin does? Pepsin goes and cleaves the amino terminal, that is amino terminal here, amino terminal of the aromatic amino acids. As a result, suppose there was one phenylalanine in this we can say protein. So what happens? It gets cleaved into two, that is long polypeptides, they are cleaved into a mixture of smaller peptides. Okay, after the digestion completion in the stomach, the food contents of the stomach, it enters the small intestine. Due to the acidity of the stomach contents are acidic in nature. So when this acidic stomach contents enter the small intestine of the digestive tract, this acidity, or we can say the low pH, it triggers the release of a hormone termed as secretin into the blood. This secretin hormone, which is released in the blood, it stimulates pancreas to release bicarbonate ions, which neutralizes this acidity of the stomach contents. Once the amino acids reach the small intestine, these, we can say, stimulates the release of hormone cholecystokinin in the blood. Cholecystokinin in turn, it activates, or we can say stimulates, pancreas to release several enzymes in their zymogenic form. By zymogenic form of enzymes, here I mean that they are inactivated forms of enzymes. All the enzymes which are released by the pancreas, they have optimum pH in the range of 7 to 8. Now let us a little bit discuss about the pancreas. It is an organ of digestive and endocrine system of vertebrates. It has both endocrine and exocrine function. It is the only organ in the human body which has both the functions, an endocrine function as well as an exocrine function. Alright here, but the most important is the exocrine function of the pancreas. The exocrine cells of the pancreas secretes various enzymes in the zymogenic forms. The enzymes are trypsinogen, chymotrypsinogen, procarboxypeptidase and proelastase. Let us discuss about the first enzyme which is released by the pancreas in the small intestine to aid the digestion of the proteins in the small intestine. Trypsinogen. Trypsinogen is activated to trypsin by an enzyme which is termed as enteropeptidase. This enzyme, enteropeptidase, is released by the intestinal cells of the small intestine. Alright. On one hand, it is activated by the enzyme, we can say enzyme that is enteropeptidase. On the other hand, it is also activated by another phenomenon which is termed as autocatalytic cleavage. I already told you what is autocatalytic means. What does this mean? That is, it once activated trypsin, it goes and activates its own zymogenic form, trypsinogen to trypsin. Alright, it is activated by enteropeptidase and autocatalytic cleavage. Once the trypsin is activated, it goes and we can say activates the other enzymes which are released by the pancreas. Trypsin goes and activates chymotrypsinogen to chymotrypsin, procarboxypeptidase to carboxypeptidase, and last, that is proelastase to elastase. Now here the question comes, why are all enzymes released in their zymogenic forms? All the enzymes are released in their zymogenic forms because if they are released in the activated states, it will destroy the cells from which they are synthesized, or we can say which from they are excreted. For example, let us take an example that if we can say trypsin from pancreas is secreted in the trypsin form, not in the trypsinogen form, what happens? Trypsin will go and it will cleave, it will hydrolyze the various proteins which is present in the pancreas and resulting in the damage of the organ from which it is secreting. Besides the secretion of the enzymes in the zymogenic forms from the pancreas, another factor, we can see another protein is synthesized in the pancreatic exocrine cells, that is pancreatic trypsin inhibitor. We already took a look that trypsin is the main enzyme. If it gets activated, it will activate all the other enzymes. So there is an inhibitor which is released along with the four enzymes which prevents the activation of the trypsin. Thank you so much for watching my video. Please like, subscribe and share.